how payers treat these tests, or you think how consumers adopt them change if it was FDA regulated versus the current construct, where they're basically looked at by CLIA?
Sure. Frankly, I think today payers are not terribly concerned about the approval process itself. If the FDA says that a company can't market a test without FDA approval, it's a done deal. We won't cover the test.
Right.
But I think, frankly, today that payers are more concerned about the clinical utility of the test. How's the test going to be used? And then if there are multiple tests, which ones should we cover? So, for example, there are 3 different manufacturers of tests that look at risk of recurrent breast cancer, all using different genes, all having analytic and clinical validity studies. How do we decide which ones? How do consumers decide which ones? How do providers decide which ones are most useful in improving patient outcomes? I think that's the real challenge for us as payers today, and it's not an issue of lab-developed tests. I think the other issue, though, with lab-developed tests are there are big outs in the law that allow lab-developed tests to be used by academic institutions for their patients or by hospitals for their patients. So even if the FDA does regulate these tests, there are still a number of loopholes that will allow labs to develop their own services.
Well, loopholes presumably because a lot of these tests are being marketed as services right now, and if the FDA were to step in and try to provide uniform regulation, presumably a lot of these academic developed tests would fall under those same regulations. Now, there might be certain carve-outs for maybe a test that's promulgated only within an institution. I think that's what you're getting at, but those would be the exceptions. You mentioned the tests that do the genomic screening around predictive tests in the oncology setting. A lot of these tests are what we call multivariate tests, meaning they're looking at multiple variables, multiple genomic outputs, if you will, and proteomic outputs, and trying to discern patterns that then correlate with certain outcomes or certain predictive outcomes relative to using chemotherapy, for instance. If you do or don't use chemotherapy in a certain setting, what's the likely impact going to be, based on the ability to look at multiple variables and correlate it against some prior experimentation. Now, there's a lot of people who've long felt that these have fallen outside the CLIA framework. Can you speak a little bit to that? Because there's a distinction between these multivariate tests, which I think are a little bit more of a black box. They can't be decoupled by the physician. They can't easily be replicated by someone else who's trying to validate them versus a lot of the other tests. But FDA seems to want to regulate the whole field of laboratory developed tests.
Right, and that's the issue, is that when CLIA, remember I mentioned the year just because at that time, these multivariate tests, and I think especially since we're talking about cancer, particularly in the prediction or predictive ability for recurrence of cancers, are not as clear-cut as I think what the administrations were thinking about in 1988. And so falling outside of this is exactly, I think, the kind of tension spot, which is why I mentioned cancer testing, autism spectrum disorder testing, which rely on those variables. That is, I think, to be honest with you, going to be the heart of where the FDA is going to have to make some decisions about, is this their wide regulatory reach? And then what is that going to do to the marketplace? Right now, one of the reasons there's such success of these LDTs is their consumer ability as well as the utility. Physicians have now got a whole kind of armament, so to speak, of testing that they can offer patients. And honestly, sometimes even out of pocket, they're at a lower price point because of what's kind of fallen outside of the regulatory process, which tends to bring on administrative costs, et cetera. So I think we're talking about a tension of how fast, how soon to market, and what really is the market, and what is the reach for the administration.
Agrees that it would increase the costs and slow the introduction of these tests into the market. What do you think? What do you expect to happen here?
I expect the administration has. Let's be honest. There's a lot. I mentioned the media because there's been a lot of publicity around certain companies and certain tests in particular. I think that's going to, I think the report that the FDA released already put in an opening salvo that, yes, we're going to be looking at this closer, and I think the FDA is going to be pressured to take action on it.
But I think there are some fundamental differences in the different types of tests. If you have single gene assays, establishing analytic and clinical validity is much easier, or if you have multiple gene assays, it's when you get into whole exome sequencing or whole genome sequencing, when you're identifying 3.5 million mutations or variations in a single patient. What does that mean? How do you establish clinical validity when you have no idea what these mutations signify? identify. They may be rare or novel mutations, and I think that's where we're going to have tremendous trouble establishing validity. What we really need is a database that we don't have today that will help us identify rare or novel mutations that are associated with disease. And that's my big concern right now, is that we're going to be generating a lot of data and we won't know what to do with it because we don't have a single library of data that we can learn from.
Presumably the FDA regulation, if they implement it the way they're talking about, isn't going to satisfy the needs that you're talking about either, because the FDA says that they're not going to get into clinical utility for the vast majority of these tests.
They're going to use established databases, right.
Or they're just going to certify that if the test purports to measure a certain output, it actually measures that output, and they're not going to get into the question of is that output meaningful or not. Right. That would be clinical utility, and they've said that they don't want to get into regulating those kinds of decisions. They want to leave that to the physician to interpret the information.
But even in terms of a database, the president's initiative has a goal of creating a library of 1 million different genome patterns, and that's not enough, I don't think, especially for rare mutations. So I think the FDA, I think payers like the fact that it's in the market because there are a lot of claims being made, but the biggest challenge will be how do we establish the clinical validity and utility of these new findings.